You heard already a really nice overview of molecular and cell biology, so my task was to try to give you a little bit of a feel of an overview of mechanics. And I understand from this morning that about 25% of you or so are coming from a life science background, so the apology is you're going to see some things that you maybe haven't seen before. But I think that's okay. In fact, professionally, I enjoy working with people who know things I don't, because then I learn something from them. And I think that this field, by its very nature, not only encourages, but requires us to interact across disciplines, and I've enjoyed that over the years. So what I want to do is give you more of an outline of the mechanics that would be used at a tissue level. And I want to put that into a context and really make it in an outline form, hopefully that's easy to remember at least the bits and pieces. For some of you that you would be able to really master it and, or grasp it, and others just so that you're aware of it, so that when you talk to people who are engineers or physicists or mathematicians, that you sort of know at least a little bit of the language and why they're saying what they're saying. Okay? So what is mechanics? Can somebody give me a definition of mechanics? It's the study of forces and motions. That's great. So you can write that down. That's the classical definition of mechanics, the study of forces and the associated motions. And once you've written it down, you can say, but Jay said that he would like to broaden it. Because I really think it's much more than just forces. It's really applied loads. And those applied loads can be chemical loads, electrical loads, mechanical loads, which we call forces. So mechanics to me is really the study of applied loads and the responses of the material. So what is biomechanics? It's the study of the response of living things to applied loads. And then once you have that written down, I'd like for you to say, but Jay said to modify that, because when you say applying mechanics to living systems, I think it really limits what we can and should do. Because to apply something necessarily suggests that there's a knowledge base that you can take and simply apply. And much of mechanics was developed not for biology. It was developed for structural engineering, civil engineering, mechanical engineering, etc. So I really think that a better definition of biomechanics is the development, the extension, and yes, application of mechanics to understanding living systems. Biology, pathobiology, physiology, pathophysiology, etc. So my subtle distinction is it's the development, extension, and application, which really means that you want to be careful not to go and just take what you know from mechanics and simply apply it because it may be misapplied. It maybe was not intended or developed for that particular point, okay? So mechanics is really the study of applied loads and the responses by the material. And in biomechanics, we need to sometimes extend and develop that mechanics. Why is mechanics important in biology? And at the end of the prior session, the question and answer, we had some hint to that. Why is mechanics important in biology?
Coordination.
For what?
For coordinations.
Coordination of movement. So just what I'm doing now. Okay. What else? Absolutely. So biological tissues and cells respond to mechanical signals. So I would say that's one of the two key reasons why mechanics is important. The first is simply that many tissues, remember this is tissue mechanics, many tissues, bone, muscle, ligaments, tendons, etc., their primary role is structural. Their primary role is mechanical. So biomechanics is important both because of the structural role and the fact that many cells respond to mechanical stimuli. And I really want to say mechanical stimuli rather than forces or applied loads because the stimuli could be come in many different forms. In fact, if we take this second reason and look at it a little bit more carefully, you can imagine the fundamental unit of life, the cell, and basically say that if you have a mechanical stimulus, in many cases, what do you get out? You could get a chemical response, or biochemical response, or I'll just say biological response, which in many cases is going to be chemically, have a chemical underpinning. So you get a biological response. And as Roger said earlier in the question and answer, many cells in the body actually exhibit this type of response to mechanical stimulus. In fact, this sort of paradigm has given rise to a term called mechanobiology, just to emphasize the importance of the mechanical stimulus giving rise to a biological response. Roger also mentioned that a prior GEM 4 meeting was developed not around or organized around developmental biomechanics, but actually mechanotransduction. And in the early days, really what one would put in the middle here is the term mechanotransduction. But as you heard earlier this morning, there's mechanotransduction. We heard a lot about transcription. And then finally, translation. So this mechanical stimulus can give rise to multiple responses at the cell level. Literally, the sensing of the signal, turning that signal into some particular information from the DNA as far as transcribing certain genes or expressing certain genes, and then finally turning that into a protein, for example, response. The mechanics comes in because how do you know what this stimulus is? Well, you need to know mechanics in order to understand well what that stimulus is. Some of these biological responses may give rise to a change in what? At a tissue level, maybe the geometry of the tissue. Maybe the properties of the tissue. Maybe even the load that the tissue is exerting on the cell or vice versa. So when you think about these biological responses, they can lead to, they don't necessarily, changes in three things: geometry, material properties, and applied loads. And if you want to define a mechanics problem, you need to do what? Define your geometry, your material properties, and your applied loads. So there's obviously this interplay. So mechanics is important for many reasons. So what I'd like to do today is go through and give you a very brief introduction in a way that I hope is somewhat memorable with regard to mechanics. And I have a hard time remembering things, which is why I write a lot of things down so I don't have to remember them. But in mechanics, I think what I need to remember is to basically remember 5. And you'll see this repeated, okay? So 5 things. So as far as mechanics, I really think there are 5 areas of mechanics. When you think of mechanics, who's the first person that comes to mind? Newton. So Newton really developed mechanics that we would now refer to as a discrete mechanics. And what do I mean by discrete? Whether he was looking at the apple that was falling from the tree or looking at the moon at night, he imagined those objects as mass points, okay? As discrete entities, mass points. And of course, one of Newton's famous equations was what? F equals ma. So he's really looking at mass points, discrete objects. One of the most prolific mathematicians of all time, Leonard Euler, and subsequently Cauchy, introduced what we would now refer to as continuum mechanics. And this is what we'll emphasize today because Roger asked me to talk about tissue mechanics. And tissue mechanics is typically looked at within the context of continuum mechanics. But to put it all in perspective, we also have statistical mechanics. We have quantum mechanics. And we have relativistic. So there are really 5 areas of mechanics that really form much of physics. We're going to focus on continuum mechanics, but I think, and I don't know what Roger would say, but I think that most of mechanics today in biomechanics focuses on these 3 levels: discrete, continuum, and statistical. Discrete could be if you're interested in gait or athletic performance. The Olympics are coming up in London, looking at the mechanics of somebody getting over a bar as a high jumper or a pole vaulter. You could use discrete mechanics and look at what happens to their center of mass. Statistical mechanics, maybe you're interested in polymer mechanics, and a biopolymer being actin, etc. But again, we're going to focus on continuum mechanics, okay? So when you talk about continuum mechanics, the first thing is, when can you use it? When can you use it? And there's a rule of thumb that you can generally use continuum mechanics if something called delta over L is much, much less than 1. So what is delta? It's a characteristic length scale of your microstructure. So characteristic length scale of your microstructure. And what is L? It's a characteristic length scale of your physical problem. So this is a characteristic length scale of your physical problem. So let's look at a couple examples. Let's say I have an artery, and Roger mentioned my main interest is in cardiovascular mechanics, so the heart and the vasculature. So let's say that we have an artery, and we have a red blood cell, RBC, red blood cell, erythrocyte, and we're interested in the flow of this blood, which contains multiple cell types and proteins, etc. But we're interested in the flow of this blood within this artery. Can we use the ideas from continuum mechanics? Given that the length scale of the microstructure be protein or cell dimensions. So the red blood cell dimension is on the order of what? Yeah, microns, 8 microns, maybe 2 microns thick, 8 microns in diameter. So the red blood cell, the microstructure, if you will, length scale will be on the order of maybe 2 to 8 microns. In a large artery, what would be the characteristic length scale of the problem? Flow through a tube with a diameter of about, centimeters. Let's say 2 to 20 millimeters or 2 centimeters for the human aorta. Is 2 to 8 microns much smaller than 2 to 20 millimeters? That's an engineering decision. Some might say, yeah, that's small. So that's small enough for the continuum hypothesis. Let's take that same red blood cell and put it into a capillary. What's the diameter of a capillary? It's about 2 millimeters. Capillary, on the order of maybe 5 microns. So now we have to fit a 2 to 8 micron diameter cell within a 5 micron tube. And in fact, if you look at them the cells going through, they pretty much line up and take on a different geometry as they go through. So now your characteristic length scale of the microstructure is on the order of 2 to 8 microns. Characteristic length scale of the physical problem is on the order of 5 microns. So in this case, microstructure is on the order of 2 to 8 microns. What about the physical problem? It's on the order of maybe 5 microns. Is that much, much less than 1? Probably not. So you may not want to use continuum mechanics to study the motion of an individual red blood cell through a capillary. Maybe you want to use the discrete mechanics and track its center of mass, etc. Okay? Well, let's actually look inside of a cell, and I've picked red blood cells. There are other cells that are important. We talked about, so one more example. Fibroblasts, which we heard mentioned earlier. Mesenchymal cell, primary function is produce extracellular matrix, found in the skin as well as arteries in the heart, et cetera, et cetera. And if you look inside of this cell, we learned this morning about the nucleus and the DNA, et cetera. But then there are also these cytoskeletal filaments, including actin, microtubules, intermediate filaments, et cetera. So in this case, maybe you're interested in the behavior of the cell. What would be the characteristic length scale of the microstructure in this case? Would be on the order of nanometers, because the diameters of these, say, cytoskeletal filaments is on the order of nanometers. In some cases, just 1 or less. The physical problem, behavior of the cell, microns, nanometers less than microns. You can probably use continuum mechanics for some applications. So this is just meant to be a guideline. This is meant to be a guideline. Turns out that continuum mechanics behaves better than it should, which means in some cases where you would question the utility of continuum mechanics, but you use it anyway, and then you check against experimental results, you still get pretty good agreement. So the ultimate test is really the what? The experimental test. But this is a guideline. Okay? Any questions so far? Why do we use the term continuum mechanics? Essentially what we do is we introduce something called continuum averaging. So the basic idea of continuum mechanics, you getting any feedback on that? Is this a moving room? Are we okay? Thanks. So the basic idea of continuum mechanics is what we can call continuum averaging, which basically means, and you'll see something like this drawn a lot. It may represent a cell. It may represent our whole body. It may represent the heart. This is my generic continua. And so we're typically interested at what happens at a point. But really what we're doing is we're focusing in on a small volume, typically in 3 dimensions, or an area or a length in 2 or 1 dimensions. We're typically focusing on some small volume, and we're asking what on average is true about things in that volume that we can then assign at this point. So what we're really doing is we're taking a quantity of interest we're adding up everything about that quantity of interest, say, in this volume, which I'll call dV. So back to high school calculus for some of you, perhaps, the differential volume. It's a small volume. So this quantity of interest, we add it up, and maybe there's an infinity of these guys in this body. So we can add up by summing with an integral, and then divide by the volume, and we get this average. So the basic idea of continuum mechanics is you're really not interested in what's going on everywhere. You're interested in what's going on average, and you assign that average at this point. And quantities of interest typically will be things like what? What are some examples?
Elasticity.
Elasticity, which we might use the term stiffness. Yep, so that's fine. So for example, quantities of interest may be stiffness. The word elasticity really means non-dissipative. It's often misused, okay? So when you say elasticity, we're probably thinking about what is the stiffness, the elastic modulus, and that's why I put that in. Velocity, acceleration, position, displacement, stress, strain, temperature, mass density, we can go on and on. So you can put in as many of those as you want in your notes, but quantities like stress or strain or velocity or acceleration, etc. Okay? This formality is basically never done because this is a tacit assumption on which the whole theory rests. So again, to try to make it a little bit easier, how many different areas of mechanics are there in general? 5. So solving any mechanics problem within the continuum purview, there are 5 types of relationships that you always have to think about and or respect. Okay? So in continuum mechanics, always requires that we consider 5 classes of relations. Okay? Now, what I'm trying to do in an hour is what we typically do in a semester, so this is obviously abbreviated and accelerated. But what I'm going to do is give you at least a taste for these 5 relations, types of relations, and parts of them. And to do that, I'm going to try in 4 of the 5 at least to give you 5 examples. So again, it's easy to remember. Just remember 5. And if you come up with 4, you know you're still looking for at least 1. So the first type of relationship, going back to the definition, the classical definition of mechanics, what? The study of forces and the associated motions. So what might be one type of relationship that you want to look for in mechanics? What might be one thing that you want to try to quantify? Say again?
Force.
Force, absolutely. And what might be another thing that you'd want to quantify? Motions, which include accelerations and displacements and velocity. So I'm going to start off with motion. And the term that we use in mechanics, at least in continuum mechanics, is kinematics, the study of motion. So by the way, why is mechanics important? Cell migration is motion. Cell division is motion. Cell contraction is motion. Okay, and then you want to know the associated applied loads, etc. So what I want you to do is put in, if you're taking notes, 5 bullets that we're going to come back to. Okay? Measures of the motion. So somebody said the force. So we need to know something about forces. And actually, there are philosophic discussions about the concept of force, and we're not going to get into that. We're going to assume that something exists that we can call force. Some people would say, well, force is sort of a vectorial push or pull. And I already wrote down Newton's second law of motion, which is the famous what? F equals ma. Does anybody remember the third law of motion? What is it from physics? That's right. For every action, there's an equal and opposite reaction. If there is a push, there is a pull. So if a cell is contracting and pulling against its matrix, what is the matrix doing? It's pulling back. Okay? So concept of force, and I'd like for you to come in and put 5 bullets. And this, Roger, is why I asked for more than a small board. 3. F equals ma is actually one of the basic postulates of continuum mechanics and discrete mechanics, etc. It's often called a law of nature. Okay? So we need to include those types of laws or relationships or postulates that govern our behavior. And I'm going to use the word postulates. Some people refer to them as laws. And personally, I think law is a little bit of a strong term because of a guy by the name of Einstein. So these 5 basic postulates, so again, let's put in 5 bulleted areas, placeholders in your notes. But let's go ahead and go through these now. So what are some basic postulates? I already said F equals ma is a basic law. It's his Newton's second law of motion, or basic postulate. And really, what is that the description of? In physics, you didn't say F equals ma. You said we're going to study what?
Conservation of momentum.
Conservation of momentum. And in fact, there are 2 types of these conservation relations. They are linear and angular momentum. So conservation, or I would prefer to use the word balance. So balance of linear momentum. Absolutely right. And that's Newton's F equals ma, which Euler and Cauchy over a century after Newton generalized for continuum. So we have balance of linear momentum, and I'm going to go ahead and put in balance of angular momentum. What are some other basic laws in mechanics? Conservation of mass, which I would prefer to use the term balance of mass. What else?
Energy.
Energy, conservation of energy, or balance of energy. Why do I like the word balance instead of conservation of? And I've already alluded to it. I hinted. What's that? Yeah, E equals mc squared. So they need not be conserved, right? You can, but we're not going to get into that type of mechanics. But nevertheless, just to remind us that E equals mc squared is out there somewhere, and that these are not true laws of nature, but they're really good approximations under the conditions that we're interested in. So balance of energy, conservation of energy, sometimes called the first law of thermodynamics. First law. So there's maybe a second law. This one really is not a balance equation unless you have a reversible process. So this is really the entropy inequality. So again, sometimes called the second law of thermo. Wait a minute, I thought you were just talking about mechanics. Well, if we broaden our definition, it's not just the study of forces and motions, it's the study of applied loads and responses. The loads can be chemical, thermal, electrical, electromagnetic, etc. And mechanics really in its full glory encompasses many, if not most, of these things. So we have 5 basic postulates. And I've written them in a way that's a little bit funny maybe because I separated balance of linear momentum and balance of angular momentum, instead of just listing them one after the other. It turns out that the first 3 are very different than the last 2. The first 3 give rise to what we call our equations of motion. And there we keep the word motion. So you can have thermal fluctuations, etc. And these equations provide restrictions on the description of the material behavior, on the description or descriptors of material behavior. For example, heat can flow from a hot body to a cold, but not in the opposite direction, correct? The second law of thermodynamics puts that restriction. You mentioned earlier elasticity. You might think of Young's modulus, which basically says if you apply a force to a body, it will extend in the direction of the force, which means that modulus has to be what? A positive number. The second law says it has to be positive. So even though that's not thermodynamics per se, it applies in the case of mechanics. Okay, so we have 3 of our 5. Let me do the 4th. By the way, these basic postulates or laws, do they apply to, Roger mentioned earlier, no pun intended, the fluid schedule that you have, including possibly kayaking and sailing. I like that. Though the first thing I thought of, Roger's initial training was in fluid mechanics, although the breadth of his expertise is so large now. But original training was really in fluid mechanics. My original training was in solid, so when he said fluid, the first thing I thought was solid-fluid interactions. That sailboat's a solid. That kayak is a solid. So these solid-fluid interactions. These equations, balance of mass, linear momentum, etc., would they be able to describe the wind loads on the sail, which I would call a solid, and how that sail would respond? It's not a trick question. Yes. Would they be able to describe the flow of the water around the sailboat or around the rudder? Yes. So these hold regardless of whether you have a solid or a fluid, whether the temperature is high or low, within the domain of continuum mechanics. So they're very general. But we do have to get to a point where we specify. And that's where the 4th type of relation comes in. And these are called constitutive relations. Constitutive relations. Let me give you a definition here, because I think that in many ways this is the most important activity in biomechanics. And you can quote me on that. Okay, I think this really is the most important. We're really not searching anymore for new fundamental laws of nature, though some may yet reveal themselves. Concepts of force, etc., we're pretty happy with what we have, although we have generalized some of these ideas recently. Motion, pretty happy with ideas of velocity, acceleration, strain, etc. But constitutive relations, this is where a lot of activity is. These are essentially descriptions of the response of a material to applied loads, which parenthetically, which depends on the internal makeup. Let me use a different word than makeup. Internal constitution of the material under conditions of interest. And I think this last part, under conditions of interest, is really important. There are a lot of arguments in the literature. There are a lot of arguments amongst people in the community. And in many cases, they're comparing apples and oranges. One has developed a constitutive relation for one condition of interest. Someone else has developed for a different condition of interest. And they're not the same. And so they're arguing which one's better. The answer is they're both good. They're both useful under their conditions of interest. And an easy way to remember that is, what is the constitutive equation for water? Pretty simple material, H2O. Contemplate that as you're sailing or kayaking on the river. What's the constitutive equation for water? What describes the behavior of water, the response of water to applied loads?
Hydrogen bonds.
I'm sorry?
Hydrogen bonds.
Hydrogen bonds, yeah. So that would certainly be involved. What else? But that's not a relationship. That's not a math. That's sort of a concept underlying sort of the molecular interactions. So you could talk about incompressible, maybe if you've had fluid mechanics, Newtonian behavior, any of you who are engineers. My point is that you didn't ask me the key question. It's a beautiful time of year here in Boston. That water is liquid. But when it's really cold and it turns into what? Ice, your constituent relation has to be very different because what? The conditions of interest are different. So describing even the behavior of water, you need to say, well, is it in a gaseous or steam form? Is it liquid or is it solid? Okay? And the same thing for tissues and cells. How do you find these relationships? Ultimately, you have to go into the lab. Ultimately, this is one of the greatest challenges in biomechanics, and there's no answer to that question because each material, each condition, each situation is different. Nevertheless, I'm going to give you, how many points might be good to give you as a reminder? 5, okay. So, we'll give you a acrostic called de-ice. And the ice is there not to remind you about that basic example of water, but it's okay to remind you of that. Probably 20 years ago in a class, I was giving this as an example. One student said, you know, if you change this one word to this word, you'd have a nice acrostic, so I stuck with it ever since. So what are the 5 steps in finding these constituent equations? The first thing you really need to do is delineate characteristic behaviors. What do I mean by that? Is it more solid-like or fluid-like? You know, there are descriptors of cell mechanics where they really use ideas from fluid mechanics. They say maybe in the migration of the cell, I can treat it in somewhat of a fluid-like manner, or maybe in morphogenesis. Whereas in other cases, it's solid-like, or maybe there are cases where it's both. Solid, fluid. We use terms like viscoelasticity, viscous and elasticity. Is it elastic? Which means any energy that you store in it, it gives you back. It's non-dissipative. Or is it inelastic? Word we often use for inelastic is something like plastic. If you take a plastic milk carton and you deform it, it doesn't come back to where it was. Okay? And so it's not, all the energy is not recovered. Some of it went into some changes at the molecular level, et cetera, et cetera, et cetera. Linear, nonlinear, incompressible, compressible. So once you've delineated these characteristic behaviors, then you're in a position to establish a theoretical framework. And again, the word elasticity was used earlier. There are theories of elasticity. There's a theory of linearized elasticity or linear elasticity. There's a theory of nonlinear elasticity. There's a theory for plasticity and viscoelasticity and Newtonian behaviors, etc. So you figure out how this material behaves, which means you sort of classify it. And once you've classified it, you've sort of defined something. And you say this theory will describe the behavior of things that fall into that class. And then once you know this theoretical framework, you need to identify specific functional relationships. For example, in the theory of elasticity, you might say, well, I want to relate something I call stress to something I call strain. Fine. But do you do that functionally in a linear way? If it's a metal or bone, you probably do, or a bio implant, titanium, etc., a stent, intravascular stent. Or is it a nonlinear relationship? Maybe it's a tendon or ligament or artery or the lung or a cell. So you need to identify those functional relationships. Of course, those functional relationships will probably have some parameters in them. which we often call material properties or material parameters. So we'll have to calculate the values of these parameters. For example, you may have fluids like synovial fluid or maybe plasma or cerebrospinal fluid, and you may use the same functional relationship, but they may have different material properties. They may respond to load differently. They may have different viscoelasticity or viscosity, resistance to flow. And then finally at the end, probably the most important thing, the thing that we tend not to do, is to evaluate, well, how good is this relationship, which was determined often in the laboratory, at least in part, under specific circumstances. Does it have predictive value? If it doesn't, then all you really need is the original table of data. You don't need to have an equation that describes it per se. So you want something that you can evaluate predictive capability. Okay? So if you are charged with finding a new relationship that describes mathematically the behavior of a certain material, tissue, cell type, etc., extracellular matrix, substrate, in response to applied loads, first thing you need to tell your colleague or ask your colleague or your mentor is say, under what conditions? So I can get this manageable, tractable. And once you do that, this, I think, is a guideline of how you think through finding up new relationships. The fifth, and I want to squeeze in here, the fifth type of relation is, you can imagine that Newton, who is really the father of mechanics, what did he have to develop along the way to make the mechanics sort of work? Calculus. I mentioned that Euler and Cauchy took the discrete mechanics of Newton and gave us the continuum form. Euler is one of the most prolific mathematicians of all time. So you might imagine there's a mathematical basis for all of these ideas, and the mathematics tells us that we need to close the problem mathematically initial, which means in time, and or boundary, which means in space, conditions. Okay? So those are the 5 types of relationships we need. We need to know something about the motion, the kinematics, something about the forces. We need to know the basic laws, if you will, or postulates of nature or mechanics that control or dictate these responses. We need to individualize it for particular materials or tissues or cell types, because tendons and ligaments behave differently than the cornea, than an artery, than lung tissue, mesentery, collagen gel, fibrin gel used in tissue culture, etc. We know that there are ways to go about trying to formulate these relationships, and then we have to solve the problem mathematically, just as Newton envisioned. Okay? Any questions there as of yet? Again, I'm going through this semester course in an hour. Okay, so let me just say one thing about the mathematics. And again, we could take another semester and go through not just calculus, but something called tensor calculus. So there are really 4 types of mathematical quantities that you may deal with. The first are scalars. These are guys that don't have a sort of directionality to them. They just have a magnitude. Leave here, if you're taking notes, a little bit of space parenthetically. What are some examples of scalars that are important in mechanics? Speed, yeah, so the magnitude of velocity. Okay, so speed is a scalar.
Mass.
Mass is a scalar.
Viscosity.
Viscosity is a scalar. Although you could put that material property into a more general form if you had anisotropic behavior, but we won't get into that. How about mass density, which relates to the mass? Temperature, time. I think you get the feel that there are a few guys that we might deal with that are scalars. What about vectors? Leave parenthetically a little room. Vectors. Yeah, velocity, acceleration, force. So there we have F equals ma.
Weight.
Weight, yep, it's a type of force. Mass times by gravity. Remember, think more broadly. Heat flux, et cetera, et cetera. Then there are guys that we typically call, what's next in this list? Tensors. Tensors. And if anybody is interested, there's a wonderful book by Jim Simmons on, it's called A Brief on Tensor Analysis. And he says in his preface that as a young student, he heard the word in a course, tensor. He was mesmerized by it. He wanted to learn what was it. And it took him a while. He wrote this book. So if you heard the word and you don't know what it is, you can read his book by Jim Simmons and find out a nice brief on tensor analysis. So these we really haven't talked about, but things like stress and strain and others. But then there's even a higher order of mathematical extraction, which is also referred to as a tensor. And I'm going to put in here the term stiffness. And we used that word earlier, and we talked about elasticity. We used it in the first session. We talked about Dennis Disher's experiments with stem cells, putting them on substrates with different stiffness. And depending on that stiffness, you could define a lineage. Well, why did I leave parenthetically here? You can really think of, when you hear the word tensor, this is called a second order tensor. And guess what? There are first order and there are zeroth order. So if you know what a scalar is and how to use it, you really know something about tensor analysis. If you know a vector and how to use it, you know something about tensor analysis. And stiffness is actually a fourth order. But this is the guy that usually gets us. So I want to say one thing about a second order tensor. A second order tensor, and let me give it a symbol, for example, T, and you'll sometimes see this little squiggly under it that tells us it's a tensor and not maybe temperature, scalar. What does a second order tensor do? Anybody from physics or engineering? What does a second order tensor do?
It's a vector to another vector.
Beautiful. It transforms a vector into another vector. So if we have a vector here, and we have a second order tensor, it turns it into another vector. That's all it does. And you will see this type of block diagram to describe this. If we called this vector W and this vector Z and this tensor T, sometimes you'll see, and what I want you to know is that there are different notations. You might see that T takes the vector W and turns it into the vector Z. You may see that T takes the vector W and turns it into the vector Z. You may see that T takes the vector W and turns it into the vector Z. Which of these is correct? They're all correct. One is Latin, one is Greek, one is pick your language. Okay? They're just different ways of expressing the same operation. But beware when you read in the literature, because you'll see papers use different notations and sometimes spend a week trying to figure out why can't I get one answer to work with the other. Okay? All right, so we're going to have to, unfortunately, do the rest a little too fast. Let's go back to this idea of the kinematics. And essentially what I want to do is I want to take my original body and draw it like this and say there's a current body. So this is maybe an artery under diastolic pressure and an artery under systolic pressure. This is maybe a cell before you've used an atomic force microscope to indent it. This is maybe a cell before it's migrating against a chemotactic gradient, et cetera, et cetera. And I want to locate a point that I want to track. Well, I don't know if you'll be able to see this very well, but here's a body, and here's a point on that body. And so if the body moves, then I want you to watch this point. So obviously it has a different position. Now if I could do this perfectly, I could get this point to this point in a lot of different ways. So just knowing the motion of one point doesn't tell me everything. It doesn't tell me if I've translated it, if I've rotated it, if I've done something else, and if I was strong enough, if I could pull it and move it over here. So what we typically do is we follow not a point, but a differential vector. Essentially the distance between 2 points. So if you want to track the motion in a cell, find 2 points in the cell and see where they go. Okay? And I'll use a capital dx and a lowercase dx to denote these two. So here's my basic math question. I want to know how to get this original position vector, which maybe if I take this guy and draw the position vector on it, and now deform it, if I stretch it or translate it or rotate it. Again, these are two fiducial markers in a cell that's migrating, two fiducial markers in a cell that's being subjected to an atomic force microscope sort of assessment of stiffness, etc. How can I get this vector from this vector? How can I get one vector to go into another vector? A tensor. And the tensor is actually called F. And that's fairly universal, although not completely so. And this F is called the deformation gradient. And it is a tensor. And next Monday, Professor Larry Tabor from Washington University is going to be here, and he may ask you if I told you about this, because I promised him I would. So we want to know how to map line elements. But you know what? We may actually want to also map areas. So not just the separation between 2 points, but maybe if I form a triad here, here, and here, then I can construct an area. So how can I map areas? So this is mapping lines. Now I want to map areas. Well, I'm not going to go through it, but each area has an orientation. So if this is an area, again on a cell or a tissue, I can define what's called an outward unit normal. And that tells me its orientation. Okay? And this outward unit normal, I'm going to call n. And this outward unit normal, I'm going to call little n. If you can get my notation, little means current and big means original. And it can be shown, which means if I had another hour and a half, that NDA equals to the determinant of F times by F inverse transpose NDA. And boy, is that a mouthful. So those of you who know the mechanics or the mathematics, you know what an inverse is. You basically, if it's a scalar, it's 1 over that guy. Transpose, if it's a matrix or it's a vector, you turn rows into columns. I'll just leave that. Determinant is a property, again, of a matrix. But what it says is that F is the fundamental measure that takes not just lines and turns lines into lines, but areas into areas. What else might you want to map? Volumes. Again, it can be shown that a differential volume, and this is where we start with our continuum assumption, is just going to be the determinant of F times by the original volume. So if you know how to map individual lines at a point, you know how to map areas and you know how to map volumes. Mechanics talks about things in space and time. So we also need to know how things change in time, which we call rates. And a rate would be something like a derivative with respect to time t. And it's helpful to know how this fundamental measure of the motion F changes with time. And it can be shown that it's LF, where L is essentially tells you something about velocity. This is called a velocity gradient. Again, for those of you who know the mechanics, this is an outline for you. For those who don't, this just tells you that when you're talking to the engineers, it makes sense that they're using these ideas. It turns out it's not really useful to know how areas change in time, but it's really useful to know how volumes change in time. And the change of a volume in time can be related to this mapping of volumes through determinant of F. So if you know how this determinant of F changes, oh boy. This is that same velocity gradient, and this just means you take part of it, for those who know the mechanics. Given time, I want to fill in these 5 guys on force, and then I'd be happy to answer any questions. So if we come over to the concept of force, one of the reasons that I drew this body with a differential line and a second differential line and an outward unit normal that basically, here's the area, and this is the outward normal. It just means it tells you the orientation of the area. One of the reasons I did that is because it is possible to look at the force acting on this guy, and we'll call that dF. So what is a force divided by an area? This is this concept of stress. Okay? But really, the concept of stress is I want to know, essentially, if I take this dF over dA, and I'm going to call it something, and I want this n. What the concept of stress really does is it tells me how to relate this force acting over this area to this outward unit normal. In other words, what I want to do is find out a way to take and transform a vector into a vector over a scalar into another vector. And I'm going to call this ratio T with a superscript n. And this is a very important guy that's actually misused a lot, or at least the term is misused a lot. It's what's called a traction, or traction vector. So how do I turn one vector into another? How do I turn one vector into another? With a linear transformation I call a tensor, and that tensor I'm going to call a T with no n. That says this traction is associated with that n. And this T is actually called the true or Cauchy stress. So I'm not going to write this down, and I'll fill in these other bullets real quickly. But a point I want to make, which is very controversial, and Professor Cam may or may not agree, I don't know. But the concept of strain comes from F. And F takes a vector and turns it into another vector. The concept of stress comes from this equation. It takes a vector, outward unit normal, and turns it into another vector we call traction. Stress and strain do not really exist. They are mathematically convenient. Consequently, can a cell sense stress or strain? I don't think it can. I think these are abstractions. What does a cell sense when you talk about mechanotransduction? Probably a force, or probably a conformational change, i.e. a motion due to that force, maybe cell matrix interactions through an integrin, which is acting on a stress fiber, etc. Does that mean that the concepts of stress and strain, which are sort of the foundations of continuum mechanics, are not useful in mechanobiology? No. Because you can derive very useful correlates. And stress and strain are things that we're very familiar with in a way measuring, at least calculating, and they're very useful correlates, even though the cell probably can't sense them. We can still develop growth and remodeling theories, theories of developmental biology, etc., using these concepts. So, does something have to be true to be useful? Can we use conservation of mass? It's not true. Can we use conservation of energy? It's not true. But it's very useful under certain circumstances. So under many circumstances, using concepts of stress and strain to correlate to the mechanobiological response, it's very useful. But let us be honest with ourselves in knowing that the cell probably cannot sense this mathematical abstraction. And if you want to argue that it can, then I'd say, well, okay, which stress and which strain, which components therein, because there's an infinity of components depending on which reference system, which coordinate system we use, etc. But they are useful nonetheless. So this is called the Cauchy stress. There are two others that are very important. The first, Piola-Kirchhoff stress, which is a force per undeformed area. This guy is really useful for experimentalists. This guy is really useful for theorists. If there's a first Piola-Kirchhoff, like if there's a first law of thermodynamics, then you can guess there is a second Piola-Kirchhoff. This one is even more interesting. Because this guy is a fictitious force divided by the undeformed area. So now I don't even have a real force. It's a force that I've operated on with F. Why did I do that? To give this tensor, this mathematical abstraction, some nice properties that are really useful in constitutive theory. So if I'm worrying about the constitutive theory and to some extent the computational mechanics, I use this guy. If I'm performing an experiment, I use this guy. It's convenient. If I'm a theorist, I use this guy. So I've got two more bullets. The beauty is that if you know any one of these three, in fact, there are other definitions as well, that they are related. So this first Piola-Kirchhoff stress is often denoted by the symbol P for Piola. And the second one is often denoted by the symbol S for second. And we can relate the Cauchy stress to the first Piola-Kirchhoff stress, which means if you know one, you know them both. And guess how we relate it? Using the same guy that mapped lengths, areas, volumes, and rates of change therein. F and 1 over the determinant of F. And the second Piola-Kirchhoff stress is related in this way. Second, Cauchy, which is called true because it's the actual force acting on the actual area. And you see this deformation. So let me summarize. I know I went a couple minutes over here, but let me summarize with a few things you don't find written in the literature. Okay? The first thing is that if you see the word stress or strain in biomechanics, or really mechanics in general, what you should ask is which one. I didn't go through it, but just as there are at least 3 important stresses, there are 3 important strains: the green strain, Almanzi strain, and linearized strain. So you need to ask which one. The second thing I would say is that things do not, quantities, theories, etc., don't have to be true to be useful. Useful means what? That you can predict something to your advantage. So these concepts of stress and strain, even though when you go into the laboratory this week and you poke on a cell, etc., with an AFM, and you'd say, well, there's a certain stress exerted on this cell, and given that mechanical stimulus, there's a certain biological response, whether it's cytoskeletal reorganization, maybe turnover of those cytoskeletal proteins, which can happen in minutes. If it's a longer-term stimulus, flow going over a monolayer of endothelial, epithelial cells, and there's a shear stress exerted on it, and it changes its production of, say, nitric oxide or endothelin-1, these vasoactive molecules through gene transcription. Does the cell sense those quantities that are easy to measure? Probably not. Are they worth measuring? Probably. And correlating that to the response. And a term that I didn't use, and we could spend another hour, is multi-scale modeling. And that's where I think you're going to ultimately connect these continuum measures, which are very important when you talk about clinical problems, which is what I deal with. Relating that all the way back to the fundamental mechanisms of, say, mechanotransduction, some of the work that Roger and others have done, where you look at cytoskeletal proteins or molecular dynamics models of, say, actin, etc. That needs to be linked. And each of those levels are important, just like investigator A has a good constituent equation and B has a good constituent equation. They're different. It's okay because they describe the behavior under different conditions of interest. Okay? So if you guys want the full semester course, come visit me at Yale. But this is all I can do today. So now I'll stop and take questions. Thank you. A 0 or a -1 that's useful in taking vector cross products. You say, well, maybe that's sort of 3 indices. We can make that a third-order tensor. It's really not a profitable thing to do. Yes?
So when you're using continuum mechanics, are there any assumptions that you commonly make, and can you speak as to how good you think they are?
Yeah, so certainly the first approximation is the continuum idea, where you say in a representative volume element, instead of knowing what's going on at every point in that volume, I'm averaging everything tacitly and then ascribing at that point the average quantity. And again, the question is that a good assumption? And I'll never forget, there was a guy visiting, it was when I was at A&M, and Wan-Muk Wong asked a question where this guy was using continuum mechanics to look at some protein folding issues that you would think, no, that should be molecular dynamics simulation. And this guy's answer was basically, the continuum theory shouldn't work, but it does. And what does he mean by that? The experimental test says that the prediction and the measurement match really nicely. So that would be one example. Other commonly made assumptions, basically in the constitutive theory, which I think is the toughest and most challenging and most rewarding part of biomechanics, is there are a lot of common assumptions. And for example, that stress depends only on strain. And I'll actually give you a good example. In a chemical reaction, let's say you have a first order reaction, you have a rate parameter. Many people call that a rate what? Constant. But it really varies with a lot of things. That rate constant or rate parameter varies certainly with temperature, perhaps pH, even stress. So a common assumption is that, no, it's a constant. Is that a good assumption? Under conditions of interest, it is, because in most cases, what are you dealing with? Isothermal problems, the body likes to stay in a fairly narrow range of temperature. So in that case, that parameter is a constant unless you have large stresses, which would change the conformation of the molecule and change its binding affinity and the reaction rates, et cetera. So those are very common, but I think that maybe the last thing I'll say as a general comment is don't believe anything I said. Test everything, and that's true with everybody this week and next. You know, you want to test these ideas for yourself. Don't just accept them. And a lot of these common assumptions are very good because they were developed and tested, and they withstand the test of time for those conditions of interest. But then you walk in and you want to use that same idea from this textbook, and it's in a new way. That common assumption may not hold. So you want to test it. Yes?
I have a related question. So going back to the guideline of delta over L,
Yeah.
much smaller than 1. So for the mechanics of DNA or protein folding, what would be a justification for using continuum?
It would not be. But again, this example I gave you was a guy who's an expert in molecular dynamic simulation who did his postdoc with Roger. Wang-Muk Wang's professor at Texas A&M. Specifically asked this question of why did you use continuum mechanics, basically rod theory, from continuum mechanics and looking at protein folding? And the guy said, shouldn't work, but it does. So in some cases, these assumptions that are commonly made work even better than what you would hope. This is sort of a rough guideline. And my main reminder there to you is I was told to talk about tissue mechanics. So if you think about an artery, it's pretty easy to accept that you can treat the artery as a continuum because the cells and, say, the collagen fibers have diameters or dimensions of microns, and the artery is millimeters thick or centimeters in diameter. But even within a cell, cytoskeletal filaments, nanometer diameter, cell micron dimensions, you can use continuum mechanics. And you can get away with it in some areas of protein folding, though I think most people would say, well, molecular dynamics would probably be a better approach in general. Roger's the expert there.
Well, certainly not the expert, but I mean, it's certainly true. And there's a whole field of coarse graining molecular dynamics models, which is basically trying to bring you sort of bridge between molecular dynamics and continuum.
Right, with those types of averaging done either formally or less so. So just remember, mechanics is the subject of 5s. And if you forget, just start counting them off and find the relation that you need. Yes?
So you said that the rate of change of area was not useful. Why was that?
I'm sure that one could talk about its usefulness if you wanted to look at certain problems. But in general, in mechanics, the rate of change of a length is very important. It's called stretching. It's fundamental, deriving even the continuum mass balance equation. Rates of change of volume are just very important because everything is volume averaged. Areas, you just don't see it a lot. But if you did some two-dimensional theories, worried about maybe cells that preserve or don't preserve cell membrane surface area under certain types of loads and how that would change dynamically. You know, and that's why I said the important thing to remember is mechanics is not biomechanics is not the application of mechanics to biology. Because then you would look in the book and say, well, nobody's looked at the rate of change of area, so it must not be useful. It's the development extension. So maybe you'll discover a problem where it's important, and you'll write a paper, and the next textbook will revise it and put that in. And that could very well happen. There was a question over here or not? Yes?
Yeah, just now you mentioned that the cell, it might not sense stress or strain, but it could be a good predictor of something happening. So there's been studies showing that, for example, the cells are sensing rigidity or deformation of the substrate. So which of these do you think is a better descriptor or predictor?
Again, I think it really comes down to the conditions of interest and the question of concern. And so in that case, you may be interested in what is the substrate stiffness and correlate stem cell differentiation according to stiffness. But obviously, as the cell is sort of assessing that substrate, it's pulling on the matrix is pulling back. So there is some force-like or stress-like quantity. It's deforming the matrix as it's sort of probing it. So there is some deformation. So you could use deformation or strain, you could use stress, you could use stiffness. Which one is better? I'd say that's really, the answer is the experimental test. Try to correlate biological response, whatever you're interested in, whether it be gene expression or motility or whatever, versus these different metrics and find out which one is most reliable and consistent. And if that is sufficient for your subsequent predictions, that's a good thing to do. A good example is an endothelial cell in an artery. So it's subjected to an artery, it's subjected to this pulsatile fluid. Well, as the fluid is moving, the cell surface moves. Some people believe there's a glycocalyx surrounds it, some people don't. Whether you believe it or not, the cell will sense in some way that motion. Would you want to correlate its subsequent gene expression, maybe production or upregulation of nitric oxide, or endothelin-1, or Ang2, or a list of all these vasoactive or growth promoting or whatever molecules to that deformation? You certainly could. Nobody does that. Because in order to do that, you know the force in the fluid, or if you will, the shear stress. But you would have to know the constitutive properties of the cell, and in particular the cell membrane of the glycocalyx, if it exists, to calculate the deformation response. Nobody's confident with that. So what do? They correlate the shear stress because Newton said for every action, there's an equal and opposite reaction. So the cell must be sensing, really experiencing that shear stress. So you correlate nitric oxide production with shear stress, not deformation. Conversely, you can take a smooth muscle cell or fibroblast, put it on a flexible membrane, and stretch it. Well, as you stretch it and it deforms, is it sensing some force? Yeah, I mean, there's some force that's loading it. Is there an associated stress? Yes, you can calculate any of the three. Is that easy to do? No, because you need to know the constitutive properties of the cell membrane, the cell matrix interactions, etc. But what is easy to correlate it to? I know how much I've stretched the membrane. So they're going to correlate, say, collagen production by that fibroblast or GAG production versus what? Stress. Does that mean it's stress rather than stiffness, rather than strain? No, it means it's most convenient. And if it is predictive, I would say it is useful until we know better. And that's why you guys are here, is to learn to do things better so that we continue to push these fields forward. Yes?
Adding to his question, actually, can we say, for example, cells may feel trace of them, like pressure, which is kind of scalar now, so
yeah, and
maybe have to
that's a great question. So if the cell, let's say endothelial cell, doesn't sense the stress directly, could it sense, he mentioned the word pressure. And mathematically, pressure is minus 1/3 the trace, a term I use wherever the velocity gradient is, bottom right of the top left panel. The trace, it's the sum of the terms and the diagonal terms in the matrix representation. So pressure is just minus 1/3 the trace of the stress. Could it sense that? The trace is a coordinate invariant property. So that's nice because it means the infinity of components have all been taken out of the picture. You have a coordinate invariant metric. Maybe. You know, it really, the experimental test would tell you if you could correlate better with the minus 1/3 the trace of that quantity. So that's a good experiment for you to think about. Yes?
Could it be that cells are, so instead of sensing the mechanical loads, it's sensing its own responses to that, like its own deformation?
Oh, absolutely. I think that fundamentally, and I'd love to hear Roger's comments, fundamentally, at an atomic level or molecular level, there are forces, certainly, and there are conformational changes of these molecules. So do you want to look at that in terms of entropy or literally the deformation of the molecules? What's the best metric? I don't know. But certainly that is what's happening, and I think that's what's happening. That's what the cell is sensing. And these conformational changes of these molecules can change binding sites for these second messengers and phosphorylation and all of these things. So I think the conformation of the molecule makes obviously a big difference, changes these rate parameters, which in turn is being driven by the force through Newton's third law, which you can correlate to stress or traction or trace of that, etc.
In
Right.
all these equations, we just said that we just select one area. So if I select one means condition of interest, there is possibility I select by mistaken I select the wrong area of interest, so my whole experiment is gone. So is
Yes.
there any criteria to just select this one for this experiment, this one for this experiment? Means we just think about different parameter, mass, momentum, everything. So
That's a great point. And continuum mechanics, part of the name is continuum, which means that at every point in the body I can define all of these things. We don't yet have a Heisenberg's uncertainty principle that I'm aware of in biomechanics, but certainly something like that's going on.
because
And-
when
and-
I worked in flow cytometry,
Yes, please.
Please interrupt. When I worked on in flow cytometry, so for that, sometimes it happened that you select a wrong population, and sometimes it happened that area not give you the good result. You have to select the height and width parameters for that. So
Yeah, and this is what's so difficult about biomechanics. So I hear that they're going to do an AFM experiment next week. So atomic force microscope is basically a very sensitive profilometer. So you're going to come down and indent a surface. And let's say you're interested in measuring elasticity or stiffness of the cell. As soon as you indent that cell, you change the mechanical loads on that cell, you can have within minutes cytoskeletal rearrangements. So that what you're measuring is actually being changed by the measurement instrument. So when you finish the experiment, do you really know what you set out to know? What you know is how the cell responded to the experiment locally. So how can you assess stiffness somewhere else? Well, you go over there with the probe and you do it again, but you have the same problem. So we need to keep in mind that in biology, whether it's in a cell culture system, a tissue culture, and we put cells in fibrin gels versus collagen gels with fibronectin or vitronectin or whatever, that experimental condition will dictate, in many cases, monolayer versus three-dimensional matrix will dictate in part the cell's response. And we have to remember that we're measuring the response of the cell to its environment. And we've changed the environment. So what you try to do is a lot of different experiments so that you have different classes of environments and you start looking for consistent behaviors. And then you can extrapolate or predict. So just like in the acrostic de-ice, you always have to evaluate the predictive capability. Because you don't want to know just how does your cell respond to this rate of AFM indentation, this depth, in this culture media with this cell on this substrate. Because that's not particularly helpful. How can it be generalized?
So if my instrument can measure the change or the response in height-wise and in area-wise, and I get the response in height-wise and not the area-wise, how I can interpret that result? How in this?
Yeah, so again, that's the experimental challenge. I can't answer. I mean, that's a good sort of.
How can any theory will explain my result? If I get the response, my instrument can measure height-wise and area-wise. I get the response in height-wise, not the area. In terms of area, my cell is like stuck up. But in terms of height, I get some response though. So how can I interpret with this area?
It all depends. So if you're making a local measurement, then hopefully some of these other factors is sort of dimensional similarity, that you get away from some of these dimensions because you're really trying to assess properties, behaviors, et cetera, locally. But we can talk more about that. I'm actually staying for lunch, I think.
Yeah.
And
I wanted to set the stage too for. Jay really is the expert on growth and remodeling and how you use this kind of an approach, a continuum approach, to describe growth. And since we're going to be talking a lot about growth and development and also remodeling of tissues, those are some things. I'm sure there's no short answer that you can give that would sort of describe how you build growth into this sort of general approach. But those are the kinds of things that I think might be interesting to discuss over lunch.